but it's also one of the most complicated aspects of immunology, which is signal transduction. And a lot of people see signal transduction, they see all these different molecules involved, and they kind of like get all this panic of seeing too many molecules and what they do. So what I really try to do is take you through this process at a very step-by-step conceptual approach, and then at the end fill in the details of how it works. So the questions to consider is basically, after recognition of its cognate MHC peptide, how does the T cell receptor activate immune response genes? The T cell has to turn on genes within the T cell in order to have its effector functions. How does that process occur? What's involved in that process? What are the structural motifs that are used by signal transduction molecules that allow specific interaction and activation of effector proteins? And this is the most important question. The nucleus, where the chromosomes and genetic information are, is inside this nuclear envelope. It's inside this bubble. Outside, all sorts of stuff is going on, infections, terrible things, and yet inside this nucleus bubble, the genes may have no idea what's going on outside. How does that occur? Because at the end of the day, the genes have to be turned on to make whatever needs to be made in response to the outside environment. Yet how does the genes know what's going on in the outside environment? That's really the critical question that is involved in terms of signal transduction process. So before we kind of get into T cell receptors, I first want to provide some general signal transduction concepts. And again, a lot of you probably are familiar with this, but again, as I've learned, there's no one ever is against reviewing some of these concepts. The first concept to appreciate vis-a-vis the T cell specific response, and this we're going to expand upon it more in lectures tomorrow, is that it's a multiple signal process. So the paradigmatic class 2, in this case MHC peptide, T cell receptor, CD4 interaction, the same thing would be the case for CD8 interaction. That provides the initial antigen-specific signal. However, that signal is not enough to turn on the T cell. The T cell needs, in addition, a second signal, and I'll be discussing a little bit, maybe some detail today and a lot more tomorrow, exactly what's involved in that second signal process. But the question that I want to address that I'll answer today is, how do you integrate two signals? And what happens is, if you get one signal, nothing happens, and in fact the T cell gets turned off. If you get this second signal in the absence of the antigen-specific signal, nothing happens. Only when the T cell gets two signals is the T cell activated to make the appropriate response. Well, why is that? How could two signals integrate at the genetic level to allow an immune response to be generated? And conceptually, what is the reason for having a two-signal system? So the example that I give in the States is, which may be applicable here, is a safety deposit box. So did anyone here ever use a safety deposit box? Anybody? You don't have safety deposit boxes here? Well, in America, we're very afraid of crime. So people take all their valuable stuff and bring them to the bank in this giant bank vault, and they rent a box. And they're given a key to use for their specific box, so nobody else can get into the box. However, for some reason, the bank doesn't trust you. They think in the middle of the night you'll break in and try to get to your box without them knowing it, so they also have a key. So in order to get into a safety deposit box, you have to insert your key into your specific box, and the bank manager puts their specific key into their keyhole, and when both of you turn it at the same time, then you can take your box out. So that's how it works. The reason is, clearly, the bank can't get into the box without you. You can't get into it without the bank, and that's the protection mechanism the bank utilizes. Cells do the same thing by having this kind of two-signal process. You have this specific signal that's your key to your specific box that's unique from person to person, and this signal is unique for MHC peptide to TCR. But then there's this generic second signal, which represents the key the bank officer uses, that's the same for every box, but unless that's turned, no one can get their safety deposit box. So this combination of a specific sequence and this generic sequence allows you now to really give you an extra level of control in terms of turning the T cell on. And again, tomorrow I'll explain why actually turning off a T cell when it only sees one signal actually can be a good thing in terms of eliminating self-reactive T cells. Okay, so just keep in mind, two signals, how can we explain this at the molecular level? Now, there are some cellular receptors. So now this is extramembrane, this is inside the cell, this is a receptor. These, in this particular case, would be a homodimer because these proteins are exactly the same. When they're separate, their kinase domains cannot interact with each other and phosphorylate each other. And again, the example I always think about is, anybody have kids here? More than one kid? Okay, how many kids do you have? Two. How old are they?
4 and 7.
4 and 7. Do they ever fight with each other?
Yes.
They do. Okay. When they fight with each other, how do you resolve the fights?
Generally shut both of them out.
But when your wife's around, you have to do the right thing. What do? You know, act like a parent.
Trying. I'm not sure what the right answer is. Well, you find out what the problem is, I guess.
Oh, but I mean, like, let's say you have to work and you want them to be quiet very, very quickly. How do you stop the fights? You separate them, right? You know, you go in that room, you go in that room, and then we'll deal with this later when your mother comes home, right?
That's probably what she does.
Yeah, oh yeah, until you come home, and Daddy will take care of you. So the same way with receptor molecules. If you don't want any transduction to occur, you separate those molecules. They're not doing anything. However, if now, let's say you want them to interact, what do? You bring them together. And what's going to bring them together is whatever the ligand that they bind to. Now the ligand comes, it brings the two receptor chains together because it's now forms a dimer. But now these kinase domains, when they're separate, which couldn't interact with each other, now can phosphorylate the substrate, which is present in the opposite chain, and therefore start inducing a signal transduction pathway. But in this case, the kinase domain is an integral part of the receptor itself. Okay, so that's a very simple, straightforward transduction pathway. However, other cellular receptors are transduction molecule challenged, the politically correct way of saying that they lack kinase domains. And even though the kinase domain is not part of the receptor protein itself, they have associated with them physically a kinase protein that is now the same concept. When they're separate, no interaction, no phosphorylation, no signal transduction. When now the ligand comes, brings these two molecules together, the kinase protein that's associated with the receptors now interact with each other, phosphorylate each other, and now cause signal transduction. So that concept of basically bringing two chains together is one that's utilized by many signal transduction pathways, and either the receptor has its own integral kinase or it has to have a kinase molecule associated with it through some other interaction. Okay. Now, why do we have phosphorylation events? And the other major concept in signal transduction is you need to amplify the signal. So if you have one receptor, for example, or two receptors, or three receptors, and you want to turn on hundreds of genes, that's going to be very, very difficult to do unless you can mobilize an amplified signal. And what the kinases can do is kinase can amplify it dramatically. Because if you turn on one kinase molecule, then that now could phosphorylate, say, 100 substrates. If those substrates themselves are kinase molecules that by phosphorylating you're turning on, now you have 100 new molecules that can phosphorylate 100 new molecules if their kinases turn them on. And that way a single molecule, for example, can turn on thousands and thousands of different proteins down the road. And now you have thousands of proteins, in this case a nuclear transcription factor, that can bind to genes and turn them on. And that's why kinases are so potent in terms of amplifying signals and a recurring motif in signal transduction pathways. Okay, is that clear? Now, another signal approach besides phosphorylation events that's utilized in transduction pathways is calcium release. So you could basically have a receptor, it opens up a calcium channel, calcium stores that are sequestered in a certain intracellular location now get released, and these calcium ions themselves can bind to proteins, and these proteins could alter the conformational change, in this case of calmodulin, and now again cause it to activate another cascade, and again cause amplification to occur. Now, why are kinases so potent in terms of their ability to transduce a signal? And that's because kinases can change the physical structure of a protein. So for example, if I would be an enzyme and my active site would be my chest, if my three-dimensional structure was one that my arms were wrapped around my chest, would I be an active enzyme? No. However, if like my shoulder got phosphorylated and that would change my three-dimensional structure, my arms would come out and now my active site, my chest, is revealed, and now I become an active kinase. If I wanted to turn my kinase activity off, I would take the phosphorus off my shoulder using a phosphatase, and now my arms would get back to where they were and would get inactivated. And the power of phosphorylation is the ability to rapidly change structures of proteins, opening and closing active areas, and thereby turning them on and turning them off. And it's a lot more efficient than going to the nucleus, telling a gene to make a new protein, have it transported up to the membrane. This allows instantaneous changes to occur within seconds of when the signal is transmitted. It also allows you to turn it off very rapidly as well. So that fast capacity of on and off makes, and the capacity for amplification makes kinases one of the transduction molecules of choice for signal transduction. Okay, any questions? Now, in addition to being able to be turned on, as I mentioned, you also want to be able to turn off signal transduction pathways. And the way, as I had mentioned, you would turn off a kinase activated pathway is you dephosphorylate it. You bring in a phosphatase, you remove the phosphate residues, and now it's turned off. In this case, SHIP, SHP, is a well-known phosphatase involved in T-cell signal transduction. It comes on, it's like sucks off those phosphorylated moieties. Now it becomes an inactivated signal transduction molecule. The other way of doing it is a little bit more dramatic, which is basically just chop the thing up. So this is a very humane way of turning off signal transduction. No proteins are harmed in this process, but sometimes the body just says, let's just destroy it, you ubiquinate it, and now this gets processed through the proteasome, and again, very dramatic way, but you equivalently turn off the signal transduction pathway. And alternatively, it could also be destroyed in a lysosome. But for most signal transduction pathways that are rapidly being used on and off again frequently, you clearly would want to use a dephosphorylation approach. Now, in addition to the signal transduction molecule itself, you also need to have a way of recruiting molecules near the kinases, because you need to bring them into an area where they could be acted upon by the kinase. So in this example here, we have a membrane-associated protein kinase, and down here in the blue are the substrates. Well, if these substrates are down here, they really can't be phosphorylated by the kinase. It's too far apart. Well, how would you now allow these kinases to act on the substrate? You'd want to have what's called an adaptive protein. The adaptive protein has, in this case, these are phosphorylated regions here that are going to specifically bind to these substrates. It now recruits up the substrates near the membrane, and now that they're up in the membrane, they're adjacent to the kinase, and now the kinase could phosphorylate them. So these adaptive proteins, again, are a mechanism by which you can recruit substrates near kinases to allow them to be phosphorylated and then undergo whatever changes in the function they undergo. Okay, is that clear? Now, this is a cartoon of one of the major kinases that's utilized during T-cell signal transduction. And this is called a SAR family kinase. And this is first described for, obviously, SAR, but there's a whole family of kinases that have similar motifs as this paradigmatic Src kinase. And the first thing to realize is that all of these have this unique region right here, and this unique region provides specificity in terms of what membrane protein these will bind to. So as I had said before, if you have a receptor that it itself doesn't have its own kinase domain, it has to recruit a kinase to be bound to it. This domain provides the ability of the Src homology kinase to bind to a particular protein. So for example, the one used for signal transduction has a unique sequence that allows it to bind either to CD4 or to CD8 or to the T cell receptor. That's what gives the specificity in terms of what receptor it binds to. In addition, it has another sequence called SH3. And SH3 functions as an adapter protein to allow it to bind to other proteins. And the motif that the SH3 domain specifically binds to are to proline-rich motifs. So if you have a protein, it has a lot of prolines, then the SH3 domain will bind to that. The one that's more commonly used in immune signal transduction is the SH2 domain, because what makes the SH2 domain so important is that it specifically binds to phosphorylated tyrosines. And the reason that's important is if you have a tyrosine on a molecule, it can't bind this kinase, but now a kinase phosphorylates it. Now that tyrosine's phosphorylated, it's able to bind to this Src family kinase, whereas previously it was unable to. If you want to stop that molecule from binding to this kinase, then you dephosphorylate the tyrosine on that protein and it'll come off. So this SH2 will only bind to the tyrosine if it's phosphorylated. Okay, is that clear? And finally, you have the actual kinase of the Src homology kinase, the SH1. And SH stands for Src homology. So this is present in all Src families, Src homology 3, Src homology 2, and Src homology 1. Now this kinase is very interesting because it has the ability to be both positively regulated and negatively regulated, as I'll show you a little bit later. But the bottom line is that it has within it a tyrosine. So here's the kinase domain, but it also has a tyrosine, and when this tyrosine is phosphorylated, this allows this kinase to be activated. But in addition, outside of the Src homology kinase is another tyrosine, and this tyrosine, when it's phosphorylated, it turns off the kinase. And this, again, allows this kinase to be very tightly regulated by either being phosphorylated or not being phosphorylated. And again, I'll show you in a little bit in detail in a subsequent slide exactly how this works for the immune-mediated regulation. Okay, is that clear? And what's great about this kind of kinase is not only does it have its own activity, but it also has ways of binding to other proteins. It could specifically bind to some proteins that it recognizes, but it also can globally bind to a wide range of proteins as long as that protein has a phosphorylated tyrosine or that protein has a proline-rich motif. Okay, so it has a tremendous amount. It can be rather promiscuous in terms of its binding with a very specific motif. Now, another motif that's used by signal transduction molecules is, you have to think about signal transduction is getting a lot of proteins together, because when a lot of proteins are together, they can interact with each other. And if we think about our day-to-day activities, what's the most common place that people go to after work to get together? A bar, exactly. And that's nothing more than a scaffolding protein. And sure enough, this actually resembles what a bar looks like. And in fact, what happens is that when in the absence of phosphorylation events, this may be a bar that has absolutely no alcohol, so it's a bar before 5 o'clock, and then there's no one sitting here, but now there's opening time, this gets phosphorylated, and now you basically have sites that allows a large number of proteins to bind to this specific scaffold protein, and now sitting together they can all interact with each other. Now think about this. If this, for example, were tyrosine residues, when they're unphosphorylated, SH2 domains don't bind to it. However, if you now phosphorylate a bunch of tyrosine residues in this particular protein, now you can have different proteins with different structures, different activities, all of which share the SH2 domain, all binding to these phosphorylated tyrosines. If you wanted to clear the bar and get rid of these cells, what would you do? Dephosphorylate this protein, and then they come right off. Okay, so again, a rapid way of causing aggregation of proteins that you want to have interact with each other. Okay, so that's just a background of global signal transduction processes. So what I want to now kind of give you a background is just the overview of signal transduction. I'll go through it rather quickly, but then now I'll return to it step by step to go into more detail. But the reason I'm doing this is just to provide you with a broad overview before we go into the details. I think it's important to get the big picture before you now hone down and focus into each of the details. So basically, to orient you, this is the antigen presenting cell, MHC plus peptide, T cell receptor, in this case CD4, CD8 bind to the MHC molecule. That's the beginning of the process. It turns out that the CD4 molecule and the CD8 molecule, both of them have LCK is a Src homology kinase that's associated with it. When the T cell receptor MHC molecule comes into together, it recruits the CD4 protein. The CD4 protein or CD8 protein now brings LCK with it, and the LCK is able to bind sequences in the other portions of the T cell receptor. I'll discuss in more detail. This will phosphorylate tyrosines, allow recruitment of another kinase, ZAP70 through SH2 domains. This will now target adaptive proteins, activate protein kinase, phospholipase C, ultimately protein kinase, diacylglycerol, and now these will activate nuclear transcription factors, in this case NFAT, NF-kappa B, AP1 for example, and then ultimately these transcription factors will get into the nucleus and bind to target regions on genes and specifically turn on genes. So this is basically in an overview of how signal transduction at the membrane gets transported across the cytoplasm, across the nuclear membrane through these transduction molecules and to ultimately turn on specific genes. And specific transcription factors will turn on different genes, which gives you a high level of specificity. Okay, is that just the big picture? So now we'll start doing it now step by step. So the first thing to look at is that the T cell receptor is not just the alpha and beta chain. The T cell receptor is a rather complicated, large structure that includes multiple other chains. In this case, what you see is there's a delta epsilon heterodimer and there's a gamma epsilon dimer. And again, an important thing to appreciate in immunology is that a lot of multi-chain receptors are given alpha, beta, gamma, delta as a name, but an alpha, beta in a MHC class 2 molecule are totally different proteins from the alpha, beta in the T cell receptor. They're just given the same Greek letters, but it's a completely different protein. So don't get confused when you see alpha, beta this or alpha, beta that. So the alpha, beta T cell receptor chain is completely different from an alpha, beta MHC molecule, just given the same Greek letters within that receptor group. And epsilon has nothing to do with IgE, just has the same Greek letter. Okay, now if you look at this, let's look at the T cell receptor itself. This is outside the membrane, this is inside the membrane. Is there any significant intracytoplasmic protein for the T cell receptor itself? What do people think? Look at the picture. Is there a lot of intracellular protein for the T cell receptor? No, right? That clear? I mean, right? Should be very obvious, right? Okay, what is that telling you? That's telling you that the T cell receptor alpha beta chains really can't have much to do with signal transduction. Because if there's nothing sticking into the cytoplasm, there's nothing to recruit any kind of kinase domains or anything that's going to be doing signal transduction. So the alpha beta chains alone are solely going to be involved in recognizing the peptide plus MHC. They themselves will not do any signal transduction. However, that's why they have to be associated with other molecules that have domains that interact with it. In this case, the epsilon delta the gamma epsilon, and most importantly, the zeta chain. If you look at this zeta chain, it's almost all cytoplasmic and almost no extracellular. And in fact, this zeta chain turns out to be the most important signal transduction part of this complex, and it also is associated with the alpha beta. And now, if you look at this, each one of these has yellow domains. And these yellow domains are called ITAMs for immunologic tyrosine activating motifs. If you recall, when I was talking about FC receptors, I mentioned ITIMs, ITIM, that's inhibitory motifs. This is an activating motif, and as the name implies, it turns things on. So we'll discuss this in a little bit more detail, but these are tyrosines that when they get phosphorylated, they permit the signal to get transduced. So you basically have, we'll discuss in a little bit more detail, you basically have 10 of these, and these are the critical location for T-cell signal transduction to occur. Okay, any questions? So therefore, the first step in signal transduction process is bringing the kinase near the substrate. And the way that this happens for the T cells is taking advantage of the MHC restriction of CD4 class 2, CD8 class 1. Because as you recall, the CD4 molecule has a sequence in the MHC molecule that it could bind to. And when that happens, the TCR binds to the MHC molecule, but now the CD4 gets dragged alongside of it. But when it gets dragged alongside of it, it brings with it the Src homology kinase that's associated with it, in this case LCK. So now LCK, which before activation, before antigen was seen, was distal from these moieties, now it's brought adjacent to it, and when the kinase is brought near these ITAMs, these tyrosine activation motifs, it could phosphorylate these ITAMs and activate them. Once these ITAMs are phosphorylated, they're tyrosines, what type of adapter molecule can it recruit? Phosphorylated tyrosines bind to, well, SH2 domains. And again, the way of remembering it is tyrosine and 2 start with the letter T. So SH2, and in fact, this is a kinase. In this case, it's going to be ZAP70, but it has on it a SH2 domain that now allows it to specifically be recruited only when these are phosphorylated. If they're not phosphorylated, as in here, it's not going to interact. So again, the same concept. Phosphorylate tyrosines allow, in this case, SH2 domains now to bind to it, recruit other kinases to start the process. But everything is started because the CD4 is recruited to the T-cell receptor complex by virtue of CD4 binding to MHC class 2 or CD8 binding to MHC class 1, and bringing with it the LCK Src homology kinase. Is that clear? Does that make sense? So this is the Src homology kinase, and again, for immunological signaling, LCK binds to CD4 and CD8. FIN is one that I'm not going to discuss much because we know a lot more about LCK, but it seems to bind to non-phosphorylated ITAMs and just is recruited to the signal transduction location. SH3, as I'll show you, the domains are inactive when the C terminus is phosphorylated, and it's activated by dephosphorylation by phosphatases such as CD45. So now to show it in a pictorial fashion, this has been rotated 90 degrees. So instead of being this way, the molecule now is this way. Well, let's walk through the molecule. You have the unique region, in the case of LCK, binding CD4 or CD8. You have the Src3 homology domain going to bind to proline-rich areas. The SH2 domain, which can be binding to phosphorylated tyrosines. And now you have the kinase domain. Within this kinase domain, you have the activating tyrosine residue and the inhibitory tyrosine residue. When this inhibitory tyrosine residue is phosphorylated, it now becomes a phosphorylated tyrosine. What can phosphorylated tyrosines bind to? What moiety? SH2. Guess what? What do we have right over here? An SH2 domain. So now what's going to happen is this gets phosphorylated, it literally bends itself into like a molecular pretzel to bind onto itself, but in so doing, what is it covering up? It's covering up the kinase domain, so it can't work, and it's also binding to the SH2 domain, so this thing won't bind to any phosphorylated tyrosine. So in essence, it's basically turned off this protein and made it relatively not sticky. Okay? So, but now, if you want to activate this protein, you need to do two things. The first thing you need to do is you need to dephosphorylate this end tyrosine. Once this gets phosphorylated, it will no longer stick to the SH2 domain. It'll flip open, revealing this activating residue, and this activating residue can get phosphorylated by another LCK or another adjacent kinase, and now this gets activated. Dephosphorylate this, phosphorylate this, and you turn it back off again. Okay, is that clear? And it turns out that for the immune response, the critical molecule that initiates this process is the CD45 molecule. The CD45 molecule, many of you are familiar with, is the ubiquitous protein present on leukocytes, but it has a phosphatase domain, and when signal transduction occurs, the CD45 molecule gets recruited into the local area. It dephosphorylates the inhibitory tyrosine of the LCK and FIN, enabling them to be phosphorylating the activating motif, and now this was what starts the phosphorylation process going. Now the CD4, bringing the LCK that's now been activated, can now phosphorylate the targets that are present in the T-cell receptor complex. Now, as I said before, phosphorylation of receptors allows them to recruit adaptive proteins. So in this case, you now have phosphorylated tyrosines. These phosphorylated tyrosines can now bind to SH2 domains and bring in new molecules from the cytoplasm that previously were not adjacent to it. And if these molecules have kinase activity, they can either phosphorylate further residues in this particular molecule or recruit more molecules and phosphorylate them through other recruitment adapter motifs. And now you could have a situation where, again, you have new proteins, all of which are being recruited into this complex of interacting proteins that are all being phosphorylated and all being activated, again, starting this whole tyrosine process, this kinase process. Okay. So what are the roles of these ITAMs in signal transduction? Here are the ITAMs present. First of all, phosphorylation of these ITAMs allow them now to bind to SH2 domains and recruit any proteins that have this SH2 region associated with it. There are 10 ITAMs per T-cell receptor complex. In this case, you have four ITAMs with the heterodimers of epsilon delta and gamma epsilon, and there are 3 ITAMs per zeta chain, which is 6 ITAMs. So the question therefore is, why so many ITAMs? Why do you need that many? Why wouldn't one be good enough? And one first answer is, well, it's great for amplification, because one ITAM may only recruit one group of proteins, but by having 10 ITAMs, you probably recruit 10 times as many proteins. More proteins, more phosphorylation, more rapid signal transduction. Different transduction molecules bind to different ITAMs. So again, you could have a greater mix of potential signal transduction molecules there. And also it's been reported to be associated, to allow the TCR complex to associate with the actin cytoskeleton. Okay, so now what's the next step in signal transduction? You're going to need to recruit another kinase that has another group of targets that it could activate. And the critical kinase that's going to be recruited is called ZAP70. And again, this is another demonstration of why immunologists are your friends, because ZAP stands for zeta-associated protein. So what molecule in the T-cell receptor do you think ZAP is going to be associated with? Zeta, right? That's pretty straightforward. So therefore, it actually gives you the answer. So here again, MHC peptide, T-cell receptor, CD4, it's not interacting. These are the ITAMs. here is LCK, and here is ZAP70. It's nowhere near the T cell receptor because its SH2 domain looks for phosphorylated tyrosines. None of these tyrosines are phosphorylated, so ZAP70 won't associate with the ITAM. So I guess simplistically, think of it this way: if somebody wants to have a lot of friends that like them, they have to have something to offer, right? So the World Cup is coming here in June, for example, right? Anybody want to go to the World Cup? Yeah, you all want to go, right? So let's say someone you knew had 100 tickets to the World Cup. Would you also want to get to know them better? Right? Absolutely. You know, without the World Cup tickets, they were just somebody you kind of casually nodded to and said, "How you doing?" You know, whatever, and then walking away. But now your friends say, "Hey, if you need a World Cup ticket, this is the person to get to know." Next thing you know, you're baking them brownies, baking them cake, giving them cookies, etc., etc. The same way too, in the absence of phosphorylation, this is like having no World Cup tickets. No reason for the ZAP70 to hang out with it. However, once this gets phosphorylated, now it's a whole different story. Now the ZAP70 says, oh my gosh, phosphorylated tyrosine, World Cup tickets, now there's something I need to get involved with. And now the SH2 domain, the soccer homology domain, now says, I'm going to bind to that phosphorylated tyrosine, and now it all gets associates with it. That's how something like phosphorylation could really change a protein from being one that's ignored by one protein to being one that's very actively bound to by a protein. But now ZAP70 binds to the phosphorylated ITAM, in this case the zeta chain, and now it's in an environment where there are other kinases. So what those other kinases can now do is phosphorylate regions on this ZAP70 molecule and thereby activate the ZAP70. So the way ZAP70 is going to get activated, it gets recruited into this high kinase domain location, then it itself gets phosphorylated, and now ZAP70 gets turned on. Once ZAP70 gets turned on, it now starts being very active. It phosphorylates all these scaffold proteins, which before they got phosphorylated, before they got World Cup tickets, no proteins wanted to bind to it. Now they have World Cup tickets. They become incredibly popular. They recruit, in this case, GADs, SLP76, LAT. These are just ones, you know, again, clearly the details aren't as critical. And now, but what it does now recruit is a very important protein called phospholipase C. And what makes phospholipase C so important is that it has the ability to cleave, well, so aside, so this gets activated now, and this now has the ability to cleave phosphoninositol biphosphate, PIP2, into the diacylglycerol and IP3. And as many of you know, these molecules are very important because these are the molecules that activate protein kinase C. So IP3 stimulates calcium efflux, and diacylglycerol will directly activate protein kinase C. So IP3 opens calcium channels, and diacylglycerol activates protein kinase C, and now protein kinase C is going to help activate this RAS protein, and this RAS protein now is going to start a whole critical signal transduction pathway that's going to end up in a signal being transduced into the nucleus. And the way this happens is that G proteins, again, many of you are familiar with G proteins, they have kinase domains, but they're inactive whenever a small G protein is bound to it. So this G protein, GDP is bound to it, it's not active. However, if a guanine nucleotide exchange factor comes and kicks out the GDP, what it now allows is for GTP to bind to it. And when GTP binds to it, you can see the three-dimensional structure is changing, and now this becomes an activated kinase. If you basically dephosphorylate the GTP to GDP, you turn it off. So this G proteins play a critical role in signal transductions, and protein kinase C is going to be involved in turning on this RAS protein, which now is going to transduce the signal into the nucleus. Okay, any questions so far? So now we're going to start leaving the nucleus and leaving the membrane and start a signal transduction process that's going to end up into the nucleus. So again, there are multiple pathways that can transduce the signal from the cytoplasm into the nucleus. And the other important concept to realize is that different transcription factors, by virtue of their ability to bind to different regulatory regions, can activate different genes. The genes you activate is really the summation of the transcription factors that get recruited into the nucleus. So in the case of the RAS protein, which is over here, it triggers a signal transduction pathway called the MAP kinase cascade. And this consists of a relay team that transduces a signal that ultimately ends up into the nucleus. And again, this is initially very confusing, but when you kind of hone in on it, it actually makes a tremendous amount of sense. So for example, let's start with the yellow protein, which is a MAP kinase. If you would want to name a protein that phosphorylates MAP kinase, what would you call it? MAP kinase kinase. Because it phosphorylates it, then you add the kinase to it. Now, if you'd want to give a name to the protein, this is in red, that phosphorylates a MAP kinase kinase, what would you call it? MAP kinase kinase kinase. It's that simple. So here we go. We have this RAS that's been activated. It's now going to activate MAP kinase kinase kinase. Each one of these, there are different types of MAP kinase. Each one of these have different proteins, all of which has the same general motif, but have different ultimate targets. In this case, for the T-cell receptor, it's going to activate a MAP kinase 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 called RAF. RAF now is going to phosphorylate MAP kinase kinase and activate that, and then MEK, this MAP kinase, is going to phosphorylate threonine and tyrosine on MAP kinase, or ERK, and thereby activate that. So this is the MAP kinase pathway. Again, it's very straightforward. You have a RAS kinase, that's going to activate a MAP kinase kinase kinase, phosphorylates MAP kinase, phosphorylates MAP kinase kinase, then phosphorylates a MAP kinase. It's like this bucket brigade. Each one activates the one downstream of it. But again, these have same concept, but each one is different. So RAF, there can be 5 or 10 different MAP kinase kinase kinases, each one of which has a different substrate. In the case of T-cell receptors, the one that I use is RAF to MEK to ERK. Okay, is that clear? Any questions? So it's kind of a handoff. Now finally, you have to get into the nucleus. That's the end goal. It's like, you know, in soccer, you have to kick the ball into the goal. So too, in signal transduction, the nucleus and the genes is the equivalent of the goal. You have to get the signal transduction molecule from the cytoplasm across the nuclear membrane into the nucleus. And in the case of the MAP kinase, it turns out that the transduction molecule itself is ERK. When ERK is not phosphorylated, it can't leave the cytoplasm, it can't get into the nucleus. When it gets phosphorylated, now it has the ability to get into the nucleus, interact with other transcription factors, and turn on genes. So this is one mechanism by which signals get transduced from the cytoplasm, from the membrane, into the nucleus. MAP kinase pathway. Okay, is that clear? Now, a second modality for transducing signals is using a calcium pathway using a molecule called NFAT. And basically, as calcium gets secreted, it activates calcineurin. Calcineurin phosphorylates calmodulin, and calmodulin is a phosphatase. NFAT normally is phosphorylated. When NFAT is phosphorylated, it can't leave the cytoplasm. It's basically trapped in the cytoplasm. However, when it gets dephosphorylated, then by calcineurin dephosphorylates NFAT. Now NFAT without the phosphorylation can now migrate into the nucleus, look for whatever nuclear transcription factor binding site it binds to, and turn on those genes. So this is, again, how the NFAT transcription pathway works. In contrast, Another transcription pathway, which many of you are familiar with, is the NF-kappa B pathway. The NF-kappa B is regulated through a slightly different way. You have diacylglycerol activates protein kinase C, where it phosphorylates another protein called karma, which is actually a pretty cool name, and then this phosphorylates other proteins. But the end result is that it activates this IK kinase complex, and this IK kinase complex has the ability to phosphorylate IKB. Now, this is a complex of NF-kappa B and IKB. IKB is a protein that's associated with NF-kappa B. And again, if you want to be simplistic, everybody at one point had a friend when they were growing up who was really nerdy and a real loser, right? And you knew that as long as you were walking with that person and you had to bring that person with you wherever you went, you would never be able to have a social life. So what you really would want to do would be to kind of ditch that person, right? Because you knew once you ditched that person, now everything changed. Now you'd be able to be, you know, coolest person on the block. It turned out you were wrong, you the nerd, but that's another story. But the NF-kappa B has exactly the same situation. The NF-kappa B is bound to the IKB, and it really is trapped in the cytoplasm. It wants to go into the nucleus, but it can't, as long as IKB is bound. But now what happens is that the IK kinase can now come, it phosphorylates IKB. As I said before, you phosphorylate a protein, you change the three-dimensional structure, so now IKB lets go of NF-kappa B. Now that NF-kappa B and the IKB can get destroyed, now the NF-kappa B is unregulated by the IKB. It could now migrate into the nucleus and turn on whatever its target genes are. Okay, another mechanism by which signal transduction could allow a nuclear transcription factor that's trapped in the cytoplasm to migrate into the nucleus to turn on its genes. Okay, are you with me so far? Okay, so now we have to answer the question that I asked at the beginning. How can you integrate the two signals in order to allow cells to be turned on? So just to review, dendritic cells, for example, B cells, provide an antigen-specific signal, but also provide a co-stimulatory signal. How do those two signals get integrated at a molecular level? And it turns out that the co-stimulatory signal activates a parallel MAP kinase pathway that results in the transfer of a factor that requires two portions in order to be activated and turned on. So here you have the RAF-MEK, the classic antigen-specific receptor signal, and that's going to turn on a transcription factor called ELK. The MAP kinase pathway activated by co-stimulatory signals turns on a protein called June. And as shown, so basically, these you all know how this kinase works. Okay, so MAP kinase ERK activates ELK, and that turns on FOS gene transcription. So FOS is being made by the antigen-specific receptor transduction pathway. The co-stimulatory signal pathway turns on a protein called June. It turns out that FOS and June together bind to each other, form a heterodimer, and that heterodimer is a protein called AP1, and AP1 itself is a powerful transcription factor. If you just have FOS, it won't bind to the right transcription sites. If you just have June, it won't bind. Only when these two molecules form this heterodimer AP1 will it bind to AP1 regulated genes. So that's why you need both signals, because if you only turned on the antigen receptor signal, you only had FOS, that's not enough to turn on AP1 regulated genes. If you only had the co-stimulatory signal and made June, that's not enough. You need to have both signals, each one of which provides 1/2 of this heterodimeric transcription factor, and the combination of both of them, that binds to all the genes that are required to give you a real antigen-specific response. So that explains at the molecular level how two independent co-stimulatory signals can get integrated to turn on genes. Is that clear? It's a very elegant system. So, as shown in this slide here now, you have ERK, you basically now make Phos, the co-stimulatory signal makes Jun, and Phos and Jun now combine, form AP1, and AP1 binds to its target sites in genes and activates the genes. Okay? Any questions? So now we could look at the same exact picture, and whereas at the beginning of the lecture it kind of looked like this foreign roadmap, hopefully now it'll review it and now things will start to come together and make sense. So again, MHC plus peptide, first step. CD4, CD8 gets recruited depending upon class 1 or class 2 MHC. What is the CD4 molecule going to bring with it? It's going to bring with it LCK. LCK is going to get activated, not shown in this picture, but CD45 is going to dephosphorylate it. It now gets activated. It's now, since it's brought adjacent to the zeta chain shown here, it's going to phosphorylate the tyrosine residues. Those tyrosine residues now are able to recruit SH2 domain kinases such as ZAP70. ZAP70 now is going to activate protein phospholipase C, protein kinase. This is now going to increase calcium. This is going to activate calcineurin. Calcineurin is going to dephosphorylate NFAT, and now NFAT could go into the nucleus. Protein kinase C is ultimately going to activate IKK, phosphorylate IKB, allowing NF-kappa B to be released. And the two the antigen-specific MAP kinase pathway and the co-stimulatory MAP kinase pathway are both going to converge, yielding Phos and Jun, forming the AP1. And this core group of nuclear transcription factors are what going to be to turn on. So you first have the initiation of TCR-mediated signals. Now you have the biochemical intermediates. You have active enzymes. And now you have the transcription factors that can migrate into the nucleus and turn on the appropriate genes. Okay, is that clear? Semi-clear? Okay. And again, just to kind of, this is just showing the same thing in a little bit more detail. But again, the thing I want to focus on in this particular slide are the transcription factors. Because what's very, very cool about it is that different techniques are used to sequester proteins in the cytoplasm, but then allow them to be released into the nucleus. So in the case of NF-kappa B, it's physically bound to a repressor protein. If that repressor protein gets phosphorylated, it releases NF-kappa B, and NF-kappa B gets into the nucleus. NFAT, when it's phosphorylated, it stays in the cytoplasm. If you dephosphorylate it, it can get into the nucleus. And the MAP kinase itself has a target protein that gets phosphorylated. Once these get phosphorylated, they can get into the nucleus. And again, in the case of the co-stimulatory signal, they converge in the production of AP1. So these are the transcription factors playing a critical role in, for example, turning on interleukin-2 genes and a whole host of immune response genes. Because as long as you have this regulatory binding region, you could turn on the gene. So if you have 100 genes and every one of them has transcription factor binding sites for these factors, once these factors get into the nucleus, they could turn on those 100 genes. So that's how signal transduced at the membrane could turn on hundreds of genes very, very rapidly and very, very specifically. Because if you lack these transcription factor binding sites, those genes don't get turned on. Okay, is that any questions? Okay, now, you know, part of you says, like, who cares? You know, like, okay, signal transduction, very, very complicated. You know, it's never really going to be relevant. And can this do anything for treating diseases? Well, it turns out that there are two drugs that were described, Cyclosporin and Tacrolimus, which some of you may be familiar with because they're routinely used for transplant rejection treatment. Why? What are the effector molecules that are involved, cells in rejecting transplants? Take a lucky guess. T cells. Well, if you want to protect someone from having their transplants rejected, what cell would you want to block? T cells. Now, normally, in the old days, you know, like, what, 10 years ago, when we wanted to turn off the immune system, we used extremely blunt instruments. So we put patients on high-dose steroids. We put patients on drugs that had very broad immunosuppressive activity as well as having a lot of toxicity. But the idea is, let's identify a signal transduction molecule that's uniquely used by T cells. Let's block that molecule, and therefore we could specifically block T cells with minimum side effects. Okay, does that Make sense? Now, the reality of it is scientists were nowhere near as brilliant as doing it that way. Basically, you had other pharmaceutical companies that extracted fungi and all sorts of chemicals, threw them into some incredibly large screen to try to identify immunosuppressive agents. Then once they were isolated, then we figured out how they worked. But it sounds good that we designed them in a very, very proactive manner. But cyclosporin and tacrolimus work specifically by interfering with the NFAT-mediated signal transduction pathway. So normally, calcium comes in, it activates calcineurin, which is a phosphatase, which dephosphorylates NFAT, allowing NFAT to get into the nucleus. That you all know very well. Well, what happens if you block this process? And cyclosporin A and tacrolimus, working through different mechanisms, but interacting with other cellular proteins, somehow block calcineurin. And by blocking calcineurin, you no longer have the ability to dephosphorylate NFAT, and therefore NFAT can't get into the cytoplasm. If it can't get into the cytoplasm, you can't get active transcription, and therefore the T cell won't get turned on. Now, simplistically, if this is only expressed by T cells, you have a brilliant system to specifically block T cell activation. And in fact, cyclosporin and tacrolimus have revolutionized transplantation because it has allowed us to have transplant patients and do very, very well with actually relatively minimum toxicity. However, there's always a drug company that says, well, what's the market for transplant? Is it a large market? Very small market. You're not going to make a lot of money making drugs for transplantation. Let's come up with another immune-mediated disease that's relatively common that we could use our drugs to block. And in fact, a drug company came out with a cream called Protopic, and again, this is the background. In 1984, Fujisawa scientists discovered tacrolimus in a soil sample taken from Mount Tsukuba in Japan. And then tacrolimus, again, T-cell inhibitory. Well, eczema, I mean, anyone here have eczema? I mean, is it common when in Johannesburg? Yeah, so instead of using steroid creams, eczema is mediated by T cells, let's use this inhibitor specific for T cells, and in fact it works really, really well. So for a while, Protopic was actually a fairly common drug that was used, particularly on the face, because steroids actually thin the face and cause capillary generation, so they get redness. But Protopic, you didn't have that side effect, so it looked really, really good. It made a lot of sense. However, unfortunately, it turns out that the NFAT is not just plays a role in T cells, it plays a role in other cell transcription, and in fact, tacrolimus has been shown to cause increased risk of cancer. Well, for transplantation, you're willing to take the risk, because clearly kidney disease, liver disease, you're not going to survive. But something like eczema, which is not that serious, this really put a major crimp in terms of using tacrolimus clinically. But again, this is a basic immunology lecture, not a clinical lecture. Just want to underline the fact that by targeting a specific signal transduction molecule, you could cure and treat a very, very common immune-mediated disease. Okay, is that clear? Okay, so all I'm going to say briefly about B-cell signal transduction is that it's very similar to T-cell signal transduction. The same common motif. Immunoglobulin molecule functions like the T-cell receptor. When it gets cross-linked, it basically recruits other sarcomology kinases, in the case that are associated with adaptive proteins. They phosphorate the tyrosines in the associated proteins, and then these recruit SYK, which is similar to ZAP70. And again, this also stimulates a cascade of signal transduction molecules. So basically, after recognition, I think we've described how T-cell receptor activates immune response genes. The structural motifs utilize kinases, adaptive proteins, SH2 domains to allow proteins to bind. How do genes know what's going on the outside? By virtue of the fact that transcription factors that are sequestered in the cytoplasm in the absence of stimulation are allowed to migrate into across the nucleus, into the nuclear membrane, into the nucleus to activate genes specifically and turn them on, and that tells them to make the proteins at the appropriate time. Again, I know it's signal transduction is a very complicated process, but hopefully it's giving you the primary exposure to allow you to understand it clearer when you review it. Again, thanks a lot for your attention.